All right, we'll go ahead and get started. I'll share my screen. All right. Hopefully you're all able to see this. Let me know if you cannot. But welcome to the first lecture of the third annual Jacksonville Brain Bee. I just want to give you some details related to the competition, just so you know what to expect, as well as where to find any resources. So first off, what is the Brain Bee? This is a neuroscience trivia competition that was founded over 25 years ago and today spans over 40 countries as well as over 25,000 competitors annually. So this is a really large competition that was started by Dr. Norbert Myslinski from the University of Maryland. He founded the Brain Bee back in 1998, and now it's become this really large global initiative. The Brain Bee curriculum is based on standardized curriculum. It's these two textbooks shown here, BrainFacts as well as Neuroscience, Science of the Brain. You all should have access to these. They're free PDF textbooks that are linked to in our syllabus. But if you can't find them, no worries. We have a YouTube channel. We're going to post all of our recordings, lecture recordings to that YouTube channel where we'll link to the syllabus, textbooks, lecture PowerPoints. We also have Quizlets that will help supplement your learning. And that'll all be found on our YouTube channel at Jacksonville Brain Bee. And the purpose of these monthly lectures is to provide supplementary study material to aid student learning. And these Zoom lectures are given by Mayo Clinic and UNF faculty, graduate students, medical students, as well as undergraduates. The best way to prepare for the competition, however, is really to read the books. But these monthly lectures are perfect for you to come ask questions and for us to teach you the material. But we can't fit everything into a 2-hour window, so we're just going to give you a very high-level summary. But really, the best way to prepare is to read the books. We do encourage you to join these monthly lectures, however, because at the very end of the lecture, there will be a Jeopardy game, and the winner of the Jeopardy game will win a prize that will be shipped to their school. So what you learned today will be on this Jeopardy game, and we encourage you to come to the lecture. Maybe reading a little bit ahead so that you can be prepared for this Jeopardy game. So these are really fun. So how do you participate and when is the Brain Bee? To secure your spot in the competition, you should send your permission slips by November 15th to Miss Avery Hatfield as well as to myself. These permission slips, again, if you don't already have one, we will we'll attach it to the YouTube page so you can easily find it. However, if you're in our monthly email, then you should have access to it. But I mean, of course, always let us know if you can't find it, and we'll find a way to get it to you. Also, you should have a sponsor. This could be a teacher or parent who we can contact regarding details on Brain Bee location and dates. The sponsor, nothing is required of them aside from being included in this monthly email that we send out on Brain Bee updates, but they don't have to attend any meetings. They're more than welcome to attend the competition in which will take place Saturday, February 7th. They're not required to, but we do like to have an adult point of contact to give these short monthly updates. As I hinted to, our local competition, our third annual Jacksonville Brain Bee, will take place on Saturday, February 7th. All of these monthly lectures will essentially build up to that, and this will be your local competition. I'll give some details on what that will be structured like. Also, I should mention that although I have a date, it should take place on Mayo Clinic campus, but we don't have a venue yet booked as we're awaiting submission of those permission slips to ensure that we have a large enough venue booked for you all. This is why it's really important to send in your permission slips, because that's really how you secure your spot. If you want to compete, if you decide that you want to compete after November 15th, but haven't submitted your permission slip, we can't assure you that the venue will be large enough and be able to, accommodate. So please send that in by November 15th. So the actual competition will be an exam that takes place in 3 phases. Phase 1 is your typical written exam style questions that you would take in your classroom. That will be on just neuroscience facts and knowledge, some medical material, mostly multiple choice, some short answer. Then students will work their way to phase 2. This is lab exam style questions with brain models and pictures where you essentially rotate between stations to answer questions, and we'll have neuroanatomy lectures to prepare you all for this. And then the top 10 to 15 students make it to phase 3, which is a verbal competition. You will have whiteboards and be standing on stage. A neuroscientist judge will be asking you questions, and you'll have 30 seconds per question. So at the end of those 30 seconds, you flip over your whiteboard to see if you got the correct answer. This is elimination style, 3 strikes and you're out, with the last person standing wins. All students that participate and come to the competition receive certificates for participation. The top 3 students receive medals. The top school receives a prize. And the top student is sent all flight and hotel expenses paid by the Mayo Clinic belonging in neuroscience committee and neuroscience department to USA Brain Bee. So you and one guardian will be sent all flight and hotel expenses paid to the USA Brain Bee competition. And this last year took place in New Jersey. Year before that, Orlando, Florida. Before that, I believe, was California. But there's a really great opportunity, to get exposure to neuroscience and to biomedical research careers and medical careers. So that's really the structure of the competition. I'll open it for questions in a second, but I also want to highlight that we want you really to come out of this with appreciation for neuroscience and biomedical research. So on this page, you can see a variety of Mayo Clinic educational opportunities for high school students, one of them being the SPARK program. This is a program in which you can get hands-on research experience in a Mayo Clinic lab to study neuroscience, cancer, various diseases and disorders. It's a 1-year program and just really great opportunity. I believe the application deadline is sometime in December. There's just some requirements to enroll. You have to be a junior or senior in Duval or St. Johns County, and for the upcoming academic year, and have a minimum 3.5 unweighted GPA. It's a great way to get lab experience, see if it's a career that you enjoy. We also have some other volunteer programs for more healthcare and medical careers. But yeah, I'll stop sharing my screen, and if anyone has any questions, please unmute or put it in the chat. And if not, Tim will be starting off your lecture today. So yeah, thank you.
All right. Hi, everyone. My name's Tim. I'm a third year medical student at Mayo in Jacksonville. So I'm going to be going ahead and helping out with the Brain Beat program and running a lecture for you guys today. So we're going to go ahead and get started. Learning objectives. So we're going to go over the first few chapters of the book. So introduction to nervous system, neurons, action potentials, chemical messengers, senses and perception, and movement. It's a lot of material. It's a lot of ground to cover. There will be time for questions at the end if anyone ends up having questions, needs clarification, but we're going to move relatively quickly because it's a lot of material. So starting with the basics. First, just the neuroscience itself is a very broad interdisciplinary field. It covers a ton of different areas, and more or less, it's the biological study of the brain and the nervous system. And obviously, it brings in social sciences as well, math, physical sciences. There are so many different directions that you can take neuroscience with when you're trying to pursue this as a career field. So us for this, we're going to focus on the nervous system, which again is usually thought of as brain and spinal cord, but it does include all the nerves that go out to the rest of your body as well. So our brain is going to be that command center that's sending the messages, and then the spinal cord helps bring it out to your tip of your thumb, tip of your finger, all the way from top to bottom to regulate our entire bodies and help us do everything in daily life. Functional unit is going to be the neurons. So the cell for the neuron, it has multiple parts, cell body, and then it extends into this long axon. That's going to be how it communicates with other neurons. And it terminates in this axon terminal with a synapse onto the dendrites up here, or the cell body of another neuron. So overall, the just chains and chains of these together end up making our nervous system and allowing us to send signals out to our muscles and other organs with about 86 billion neurons in the human brain in total. The brain itself has some landmarks that we need to be aware of. So the largest part of the brain is what most people think of when they think of a brain, and we call that the cerebrum. So all of the normal kind of brain tissue up there is going to be considered the cerebrum. It's made up of two hemispheres. So you can see on the screen, there's a longitudinal fissure that's dividing that brain tissue into kind of two sides, the right and the left side. And those hemispheres, that longitudinal fissure completely divides them except for the corpus callosum, which is a specific structure that sits right there in the middle. It's deep in that fissure, and it connects the left side of the brain over to the right side of the brain to allow them to communicate with one another. The cerebral cortex is going to be the deeply folded layer of nerve tissue of the cerebrum. So what that means is it's literally just talking about all those kinds of folds and lines that you can see in the picture right there. That's all cortex. It's the surface level of the brain. The brain itself can be further divided into lobes. And so aside from right and left, we have four major lobes of the that are on each side of the brain. So the frontal lobe is towards the front there, and it's going to be responsible for voluntary movement. It has a role in speech, memory, emotion. It does a lot. And especially those kind of higher level cognitive skills like problem solving, personality, and more executive function. The parietal lobe is a little bit farther back and pretty high on the brain, and that's going to be responsible for integrating sensory signals from all over your body. That sense of touch is primarily located in the parietal lobe, and it can also help process some of our visual information. So we'll talk a little bit more about all of that over in the senses and perception portion of the lecture. Our next lobe is going to be that temporal lobe. So it's down on the side, close to the temple of your head, if that helps you kind of think of where it would be. And so the temporal lobe also does some of that visual processing, but it's the location for our primary auditory cortex as well. So it helps a lot with processing auditory information. And it has both the hippocampus and amygdala, which are part of the limbic system of the brain, the limbic system being how we process emotions. And they also have a role in memory and learning as well. Last lobe of the brain there is going to be in red, and that's going to be our occipital lobe. Sorry, guys. And that's going to be where we have our primary uh, cortex for sight. And so most of our visual information is going to initially be processed there in the occipital cortex of the brain. And so that's all of those lobes are part of the cerebrum, and then underneath it, this smaller kind of beach ball, volleyball looking structure, that's going to be our cerebellum, the little brain. Right. Information within the brain is going to move from neuron to neuron. So the neurons fire, they turn on, and they send that on signal to another neuron. Neurotransmitters will then diffuse across the gap and tell the next neuron in the chain of neurons to either turn on or turn off. So when we have a bunch of neurons that are all sending their axons together, and it's this big kind of bundle of just neuron cell axons, that's what we call a tract in the central nervous system. So these nerve tracts, they're nerve fibers, region spanning neurons forming distinct bundles. So the corpus callosum is a very large tract that we can see very easily when we're looking at the brain. All the neurons together end up making a neural network, which really just consists of a group of nerve tracts connecting multiple regions together within the brain. So, and these circuits help the different areas of your brain integrate information, because if you just saw something in front of you and it went to that occipital cortex and it just sat there, then it wouldn't really give us any information, because we do a lot of things moving, we respond to those visual stimuli. And so ultimately, where that information is first processed still needs to be connected to other areas like the motor cortex so that we can actually make use of all of that information. And last bit here that's not quite as related to what we've been talking about is going to be this idea of brain waves. So brain waves is really formed by this network activity, and it's what we see on an electroencephalograph, so EEG. This is something that we can use sometimes to study things like seizure disorders and just overall activity within the brain, because when a bunch of neurons fire together, that's when you can see kind of this upward spike on the graphs over here. And then as they relax, or then you get the downward spike. And so it's really not sensing individual neuron activity, but activity of these networks that we're talking about. So when a whole area of the brain is firing. So in terms of the actual waves, there are four different types of them that we can see on EEG. So alpha waves here kind of in the middle, those are the ones, they originate primarily in parietal and occipital lobes of the brain when you're relaxed, eyes closed, characterized by frequencies between 8 and 13 hertz. So if you're falling asleep during the lecture already, sitting there nice, a little bit chilling, then you're probably having some great alpha waves going on, reflective and restful. Beta waves, on the other hand, they're going to be produced by the frontal and parietal regions of the brain, ranging from 14 to 30 hertz. So these beta waves, they're a lot closer together than our more relaxed and gently awake alpha waves. Theta waves are then going to be more that drowsiness. So maybe if you've already fallen asleep, you're getting closer to theta waves, or if you're right on the cusp is really when we would see those theta waves. And then delta waves are going to be the slowest of all of the waves, which would be if you're already in deep sleep. All right, so that's about everything from that first section. So from there, we're now going to move on and talk a little bit more about the cellular level of how the nervous system works. So we're going to be looking at neurons, action potentials, and chemical messengers. So the neuron, just to review, remember it has the cell body and then the axon, which is where it sends signals out to other neurons. And then it has these dendrites around the cell body that help it receive signals from the axons of other neurons. So all neurons, they have activity that's both electrical and chemical. So neurons will receive chemical signals at their dendrites that they then convert into electricity, and they send the electricity down the axon where it's converted back into a chemical message to get to somebody else's dendrites. Neurons are a dynamic system, though. So all of the parts of the neuron are constantly in motion. So again, with this kind of electrical activity, the neuron can undergo polarization because the information is only supposed to go in one direction. And what happens with the neuron is that electrical activity, it can either be more positive or it can be more negative. And ultimately, it has to reach a certain state in order to get the electrical signal to travel down the length of the axon. How neurons then achieve this dynamic state is on the receiving side of the cell, the dendrites are in contact with axons from other cells, which is, we call that the synapse. And it's just a very, very tiny gap between those two kind of connections and projections from the neurons. Most cells in the cerebral cortex are located on the dendritic spines, and communication between those nerves is going to be synaptic transmission. So when one nerve is, or when one neuron is sending that chemical signal across the synapse to connect and communicate with another neuron, then that's synaptic transmission. So when the dendrites of that second neuron receive the chemical messenger, then it converts that into electrical currents that are much smaller than the one that goes down the axon, but they help push the neuron either towards or away from sending its own signal into the axon. So if the dendrite gets a chemical message from the previous neuron and that's an inhibitory message, it'll make the neuron a little bit more negative, a little bit less likely to send that signal out, versus there are excitatory signals that you can get as well at the dendrites, and those are going to make the neuron a little bit more positive, a little bit closer to pushing that signal down the axon to get to other neurons. So this is a little bit more about what I was talking about with the electrical state of the neuron. So normally neurons, they set pretty negative down here. And then when they get enough positivity, so when they get enough excitatory signals, they cross over this threshold. And then after the threshold, they send their signal. That's the whole depolarization phase. So that's the sending the signal down the axon. And then they have to get back, though, to this negative sense so they can get ready to send another signal. So they repolarize, but they want so badly to be at that negative 70, and it comes back down so quickly that they actually tend to undershoot. And that undershoot is what we would refer to as the refractory period. So they send the signal when it gets enough positive signals, it'll send the signal, and then it tries to get back to 70. It goes a little bit too low, and it has to bring itself back to that resting state so that it's ready to get excitatory signals and fire again. Patch clamping is one of the ways that we can study neurons and the signals that they send. So it's how a modern electrical recording technique and allows us to study which ions are moving through individual ion channels in all sorts of neurons. And it lets us make accurate measurements of these so that we can see how a neuron is firing, and when it's not, and why. So this is diagrams just showing the process there where you choose a neuron and then you form a nice seal with your pipette. You break into that membrane, and then you're now able to directly measure the electrical current that's passing through the neuron and the electrical state that the neuron is in. And that's how we then that's what we call then patch clamping, where we set the electrical state of the neuron and then we can see how it responds. Chemical messengers. So this is what we were talking about with that synaptic transmission. So neurons have specific chemicals that they will send that are typically either excitatory or inhibitory. And so from the neuron axon terminal that's sending out the chemicals, they have transporters because after you send out the chemicals, you don't really want them to just hang out there and be sending that excitation or that inhibition forever. So the transporters are what suck up extra transmitter that's just left over after they send that initial signal. And that's how that also helps then that neuron get ready to send another signal because it's now refilling its own chemical stores to be able to send that messenger again. So these chemicals, they're a lock and key kind of transmission because there are different channels for the different neurotransmitters. And so, like you can see in this picture, if the circle neurotransmitter goes and tries to activate that triangle receptor, it's not going to work, right? It doesn't fit. So it's not able to get that channel to activate. However, the triangle neurotransmitter fits in there very nicely and is going to be able to help make that shape change that will cause the receiving neuron to then change its electrical state. So a little like we were talking about with those transporters, kind of sucking back up that excess neurotransmitter in the cleft. This is a little bit of what it looks like. So initially, this is an axon up here from the sending neuron. So the electrical signal comes down, and then when it reaches this terminal, it's going to cause this vesicle filled with neurotransmitter to fuse with the membrane and release the neurotransmitters into the synaptic cleft. Once those neurotransmitters are in there, they can start to dock with their receptors that they fit in, like that lock and key kind of method. But there's a lot of neurotransmitter here, and so some of it is going to start to get sucked back upwards into the sending cell via those transporters that we talked about. And the transporters are going to let us refill the synaptic vesicles and then bring them back around so that when the axon gets another signal down here, it's ready to release the chemicals again to stimulate the dendrite of the receiving neuron. And so the action potential, what triggers all of that is the electrical charge from the action potential will change these calcium channels on the axon terminal that will open up and then bring in a bunch of calcium. And that calcium triggers the synaptic vesicles to fuse with the membrane and send out the neurotransmitters. So we have lots of different kinds of neurotransmitters in the brain that are able to trigger responses in the dendrites of other neurons. Usually our main excitatory neurotransmitter is going to be a molecule called glutamate. So that glutamate is what's going to be pushing neurons to get more of that positive charge and get closer to firing their own action potential. On the other hand, we do have inhibitory synapses as well, like we talked about, those neurons that are going to make their receiving neurons less likely to fire an action potential. And so some of those inhibitory neurotransmitters are going to be GABA and glycine. Other neurotransmitters are even more specific and they'll act on G protein coupled receptors. So instead of just triggering an immediate response, because glutamate will and GABA, they both uh, connect to ion channels that open up and they let in a big flow of other ions that change the charge of the cell. However, not all of the neurotransmitters are going to do that. Some of them will dock onto proteins. They fit with proteins that then activate other proteins within the cell, and then those proteins will go on to do things that end up changing the charge of the neuron. But it's not quite as straightforward as the simple lock and key with, it's still a lock and key situation, but it's not so much the just dock and instant change that the ion channels are. So some of these G protein coupled receptors are going to be acetylcholine, noradrenaline, and dopamine. So this is a little bit more of what I was just trying to explain with the metabotropic versus ionotropic receptors. So the simple ones are the ionotropic receptors. Those are the ion channels that we talked about. And so the ionotropic receptors, your neurotransmitter, we'll say glutamate, will come and it docks onto that receptor, and that makes the receptor open up and just inflow of ions to change the charge of the neuron itself. Now, the other option is these G protein coupled receptors, where the neurotransmitter docks and it activates this other protein within the cell. That protein then goes to an ion channel that does not have any neurotransmitter receptor, and instead the protein from inside the cell is what changes the shape of that ion channel to allow the ions to flow into the cell and change the charge of the neuron. So these G protein coupled receptors, they're a little bit more complex and they do help with neuromodulation and changing how much or how little those neurons are responding to the signals from other neurons. All right, that was all a lot. And we're going to keep going and move into senses and perception. So this is now we went from kind of overview of the brain to then going very small into just looking at the neurons. Now we're going to zoom back out even further and talk about how we perceive the world. So we have our five senses, the first being vision, and vision is obviously very important And so it has a lot of brain space that's dedicated to it. So 30% of the cerebral cortex that we talked about before is dedicated to our sense of vision. And so the pathway for how we see things is light is going to enter the eye through the cornea, pass through your pupil, and then it hits the retina in the back of the eye. At the retina, it then gets taken up by the optic nerves that carry that information back to the brain, and that information will go first to the thalamus, and then it goes back to the occipital cortex. So the function of some of those eye structures also are the iris around the pupil helps change the size of the pupil. So if it's really bright out, then we can decrease the amount of light that's coming into the eye. Whereas if it's really dark and hard to see, then that pupil opens, the iris will open the pupil wide up so that as much light as possible can come in to the eye. So that visual input is going to be mapped onto the retina as a 2D image, even though we have a 3D world. It's a 2D image because this is a flat surface, and it ends up being upside down because light that comes in through the top of the eye is going to go down and hit the bottom of the retina. So it's an upside down mapping of the world, and it's a 2D mapping of the world. So at the retina, the light is taken up. The ganglion cells in the retina will then transduce that light signal into electrical signals that go to interneurons and photoreceptors. And the ganglion cells, they each have what we would call a receptive field. So when we're looking at the world, a receptive field is going to be a space within what you're looking at and seeing in front of you that one or a collection of ganglion cells is responsible for. So if you just look straight forward and you kind of divide mentally the image in front of you into a bunch of little squares, then there's a ganglion cell in your retina that corresponds to each of those little squares that you're now pretending to see over everything. And that square would be the receptive field for that ganglion cell. The center of the retina, right here, is where our vision is sharpest. So that we call the fovea. It's a particular structure, and it has the highest density of cones out of the entire retina because our cones are those special photoreceptors that are good for two things. They see color and they help us see sharp images. They're very good at fine detail. So that's why we have tons of cones, almost no rods that are located in the fovea of the retina, because when we look directly at something, we really want to be able to see it in a lot of detail. On the other hand, as we get further out towards the edges of the retina there, we are going to have a much higher density of rods because rods are very sensitive to light, but they're not good at color and they don't sense any color and they're not good at sensing a lot of detail. So that's why your peripheral vision can be quite blurry versus whatever you're looking at is usually focused on in quite exquisite detail. The clinical correlate here is that if you have macular degeneration, you end up losing some of that sharpness of your vision because you obscure the fovea and the cones in the retina, and they're no longer able to respond to those light signals and give you the amount of detail that you want. So visual processing there's a lot that goes on with it, but ultimately what happens is those photoreceptors in the retina, they take in the light, turn it into electrical energy, it comes back and it makes a stop in the thalamus on each side, the lateral geniculate nucleus or the LGN, and then from the thalamus, that's going to go to that occipital cortex. And so the visual cortical cells, they have different types of receptive fields, similar to how we talked about receptive fields with our ganglion cells. From the occipital cortex, it's where what we call our primary cortex for sight, but it then doesn't process all of the information. So it will send some cells outwards to help process the information. So from occipital cortex, it can either go into the ventral stream, which is going to stay near those temporal lobes, and that helps us decide what an object is. So if you look at someone and you recognize their face, that's some ventral stream processing because it's what is this? Oh, it's a face, it's this person. That's ventral stream. Dorsal stream is going from occipital lobe up to that parietal lobe, and that's going to tell us where. So when you need to see like, okay, where is this thing in front of me? Where is the cup that I want to pick up and drink from? That's going to be dorsal stream processing. And our binocular vision, because it's a 2D image on the retina, is very helpful for us getting that depth perception because the brain can then compare the two slightly different 2D images that it gets and use that information to really process wear and process depth. Hearing is our next sense. So sound waves, they're going to come into the ear, go through that external auditory canal, and then they're going to vibrate the tympanic membrane or the eardrum. That eardrum is going to vibrate then three little bones within the middle ear. That's your malleus, incus, and stapes. And those bones connect to your cochlea and vibrate the round window of the cochlea and will end up transducing that message of sound, those vibrations, into electrical signals through the fluid within the cochlea that activates the hair cells in the cochlea. So, sorry, the oval window is what's vibrated by those middle ear bones. So the oval window is where those vibrations are transduced into the fluid of the cochlea, and then the fluid of the cochlea moves hair cells, and they're literally cells with what look like little hair projections on them. And those hair cells are what then turn it into electrical signals that again make a pit stop in the thalamus before they go to the auditory cortex. So almost all of our senses, before they get to the primary cortex, they're going to pit stop in the thalamus. One of the exceptions is going to be olfaction or the sense of smell. And here, kind of similar to how we had receptive fields with ganglion cells in the eye, we have what we call tonotopic mapping. And so the basilar membrane within the cochlea, it's sensitive to different frequencies along its length. So it's all curled up in here, and this spot is going to respond to a different frequency than this spot within the cochlea. Taste and smell are two of our other senses. So in the tongue, we have taste buds that are sensitive to specific chemicals, and those are going to convert that taste into electrical energy. The cranial nerves are then going to go to the thalamus again, and then they go to the cortex. So we have five main tastes that we can taste with our sense of taste. I just said taste way too many times in one sentence, but those are going to be sweet, sour, salty, bitter, and umami. Umami is that kind of savory sensation, that savory taste, often associated with meat. And so similarly to our sense of taste, there are nerves in the olfactory bulb that are going to convert odors, again, chemicals that are now in the air, directly into electrical energy. And again, this sense of smell is the one that bypasses the thalamus. It just goes straight to the cortex. Olfactory nerves are also one of the exceptions in that they're one of the few nerves that we think about that can regenerate often throughout the life. So if you end up needing more of them or they get injured and you have to make more, this is one of the times that the neurons actually behave like other cells and will regenerate over time. Taste and smell, in the end, even though taste goes to the thalamus and smell does not, they get combined in the brain and they're intricately linked to memory. Touch and pain are going to be some of our last sensations that we're going to go over. So touch and pain receptors are on the skin, and then they transmit those signals to the spinal cord, which then go up again through the thalamus, and then to the somatosensory cortex. So we have A delta fibers that are large and fast, and C fibers that are smaller and slower. And all of those are out in the skin sensing the stimuli. The homunculus is then a representation of how touch and pain are mapped out on the cortex of the brain. So this kind of figure laying over the cortex of the brain is a representation of the homunculus because down here is where we have like our foot, our toes. It's on this section of the brain. And then as we come out, it's the hand. And it looks quite strange because it's mapped disproportionately, right? So your trunk is only covering about this much space, your entire torso, really. But then that is almost the same amount of space that you're dedicating to just the fingers. And so part of that's because we have that different sensitivity on different parts of our bodies because touching things with our fingers, we need a lot more information from that to be able to really feel texture than we do on, say, our trunk. And then in terms of pain, endorphins are produced by the body to block some of that pain transmission and help reduce the sensation of pain. All right. Now, after all of that, we're going to go on to movement. So that'll be the last section of my presentation, and then we'll have another TA coming on to give you guys a further presentation on the last few chapters for this session. So movement occurs primarily, we're going to think about skeletal muscles when we're talking about voluntary movement. And those skeletal muscles are controlled by the brain and spinal cord. And so you have alpha motor neurons that run from your spinal cord directly out to those muscle cells that will then trigger the muscle cells to contract. And so, again, clinical relevance is that in ALS, amyotrophic lateral sclerosis, you're going to have degeneration of those motor neurons that then prevents you from being able to move your muscles voluntarily. Reflexes, on the other hand, are involuntary movements. They're a looped circuit that is just processed in the spinal cord. It doesn't even go up to the brain. And so there's a paired contraction and relaxation because if you try to flex your bicep and your tricep at the same time, you really can't do it. There's not a whole lot of point in that. So if you have a reflex that's triggering your biceps to flex, so then you kind of flex your arm, at the same time, it's going to send inhibitory signals out to the triceps so that the triceps relaxes and allows the arm to be flexed instead of counteracting it and trying to extend the arm. So part of that reflex is going to come from muscle spindles, where if you tap the muscle, it's going to stretch these gamma motor neurons that are involved with the muscle spindles, and that sends information back to the spinal cord about tension, stretch within the muscles, and how much something needs to be changed. Similarly, you have the Golgi tendon organs that are going to be information primarily about tension versus the spindles that are about stretch and length. All right, then we have the basal ganglia. So the basal ganglia is one of those networks that we were talking about at the start, and it's part of what helps us move. So initially movement starts in the motor cortex, and then it goes down and goes through these different areas that we refer to collectively as the basal ganglia. It's this entire network of areas within the brain. And the basal ganglia is essentially going to help us kind of coordinate and regulate our movements so that we're moving when we want to and the amounts that we want to. And conversely, it keeps us from moving when we don't want to be moving. All right. And then last bit about movement here is that you can develop movement disorders. So Parkinson's is going to be due to death of dopamine producing neurons in the substantia nigra. And this leads to loss of dopamine signaling in the basal ganglia. So if we go back right here, you have the substantia. So this is the basal ganglia, but you also have the substantia nigra that will come in and influence the activity of the basal ganglia. And when those cells die, then that's where you start to get some of the problems that you end up seeing in Parkinson disease. And that basal ganglia doesn't work as well. And so one of the things that you see is tremor, right? Moving when we don't want to, because now our basal ganglia isn't working properly. Other diseases is going to be something like Huntington's disease, which instead of losing that dopamine producing neurons, you're actually just losing, excuse me, losing the acetylcholine producing neurons in the basal ganglia. And finally, you can even just have degeneration of that cerebral cortex or of the cerebellum, excuse me. And the cerebellum is important in coordination of your muscle movements and in balance. And so when that degenerates, then you lose the ability to really do that. You become very imbalanced. You can't walk steadily. And so long-term alcohol abuse can lead to some of that degeneration or things like strokes and other conditions that can kind of selectively or non-selectively cause degeneration of the cerebellum. And that is everything I have. So do we have questions? Well, if you guys come up with any questions, feel free to throw them in the chat. If not, I think we're waiting still for our other TA to join us so we can take a little bit of a break. I
I'm
know
here
that was,
actually. But we could
oh,
take a break if you guys want. We could take a small, short break. How about a 5-minute break?
All right, so yeah, we'll do that 5-minute break, and then so we'll start up again at 5:55 then. And in the meantime, if you guys have any questions, feel free to unmute, or like I said, just go ahead and type them into the chat.
Hi guys, so I'm going to proceed and talk about the other chapters that we have. So let me share my screen. Okay, so I'm going to talk first about learning, memory, and emotions. So we are going to start off with memory. So memory is our brain's way of storing and using information. There are two main categories for memory. So we have short-term memory and long-term memory. Um, so short-term memory is temporary storage, and it holds information for a few seconds to minutes. It has three main types. So we have working memory, acoustic or auditory, verbal memory, and then iconic or visual memory. Working memory basically keeps information online while we use it, like remembering a math problem while you're solving it. We have acoustic, auditory memory, which stores sounds or words for a brief time, and then iconic memory stores visual images for just also like a short amount of time, a split second, like a snapshot. Long-term memory is much longer. It can store information for hours, days, or even a lifetime. So a quite big difference. Uh, Within long-term memory, we have declarative memory, which includes episodic and semantic memory. So semantic memory, it basically includes facts and general knowledge. So knowing, for instance, that Paris is the capital of France, that would be considered semantic memory. We also, within declarative memory, we have episodic, which is related to personal experiences, remembering those specific moments. Then we have non-declarative memory, which includes um, basically like implicit skills. So habits you perform automatically, like driving, riding a bike, all of that. And basically, the hippocampus is especially important for forming declarative memories. So one of the most famous studies involving memory includes the Henry Molaison one, where basically he had epilepsy, and in order to treat that, they removed the hippocampus and the amygdala. His seizures were improved, but he couldn't form new memories and lost memories from the 11 years before surgery. This basically taught us how essential the hippocampus is for building new long-term declarative memories. So now let's talk about how your brain processes emotions. So these emotions come from very specific brain regions working together. So first we have the insular cortex, which acts like your brain's like feeling hub. It takes signals from your body, like your heartbeat or stomach turning, and turns them into like subjective feelings. It also helps you experience disgust, which is important because it protects us from eating spoiled or poisonous food. We also have the amygdala, which plays a big role in fear, motivation, and emotional learning. It helps you figure out who's like friendly and who's a threat. It's also crucial for classical conditioning, the type of learning where your brain links a stimulus with a reward or punishment, like in Pavlov's dog's study. Finally, we have the PHE, which helps your brain modulate pain and control defensive reactions. It's involved in your stress response, reproduction, maternal attachment, and even anxiety. So you could think of it as part of your survival system. Now moving on to thinking, planning, and language. So certain parts of the brain are actually specialized to recognize specific kinds of visual information. So one of the important areas is the fusiform face area, or FFA, and it's found in the temporal lobe, and it's especially good at recognizing faces. So if you see your friend in a crowd and you recognize them instantly, this area plays a role in that. Whenever this area is damaged, people can develop prosopagnosia or face blindness, where they can see faces but they cannot recognize who they belong to. Another key area is the parahippocampal area. This region is specialized for recognizing places or scenes, like landscapes, buildings, rooms, etc. It helps you remember where you are and navigate spaces. Um, it is essentially these areas, brain regions that make sense of what you're seeing, whether it's a familiar face or place. So language is another incredible brain function. So when parts of the brain responsible for language are damaged, people can develop a disorder called aphasia. Aphasia affects how someone understands or produces language. Broca's aphasia, caused by damage to the Broca's area in the left frontal lobe. People know when they have this condition, people know what they want to say but struggle to form the words. Speech is slow and effortful, but comprehension remains intact. Wernicke's aphasia, caused by damage to the Wernicke's area in the left temporal lobe. People speak fluently, but their sentences may not make sense, and they have trouble understanding others. We also have pure word deafness, which is caused by damage to both temporal lobes. People can hear sounds but can't not link them to meaning. Like it's like constantly hearing a foreign language. So to study how the brain processes language, among other things, scientists use fMRIs, or functional magnetic resonance imaging. This tool basically helps measure brain activity by tracking the blood flow. For example, when people read or hear words, fMRI shows stronger activation in the left hemisphere of the brain, which is like the main side for language. Scientists have also discovered a gene called FOXP2 that's important for speech. It helps control how different brain areas communicate to produce the sequences of movements needed for speaking. Mutations in this gene can make it hard to coordinate mouth and jaw movements for speech. So now we're going to talk about the executive function itself. So executive functions are the high-level control processes that help you plan, make decisions, focus attention, and manage multiple things at once. You can think of it as the brain's like CEO, supervising and coordinating everything that happens. These functions take in sensory and emotional information and use that information to plan and execute actions and keep track of what's happening along the way. Um, so when you're solving a tricky problem, your brain is essentially selecting the right information, rehearsing it, and kind of like monitoring how well you're doing, all thanks to this executive function. So where does it happen? It takes place in the prefrontal cortex. That's the front part of your brain, right behind your forehead, essentially. The PFC works with many other brain regions, especially like those in the back, to process different types of information, like visual images, sounds, words, etc. One of its key job is to hold information in mind while you use it. So that's called like working memory. Like I said before, um, if you're doing like a mental math or remembering like instructions while you're walking somewhere, your prefrontal cortex is actively keeping that information active. In short, like the PFC is the brain's like control center essentially. It doesn't just store information, but helps you use that information to guide your behavior. And then one last cool idea, your brain has concept cells. They're sometimes called Jennifer Aniston neurons because they were first discovered firing only when someone saw pictures of Jennifer Aniston. So these are individual neurons in your temporal lobe that respond to very specific concepts like person, object, or idea. So for example, cells for needle, thread, sewing, and button might all be connected, helping your brain basically group ideas together very quickly. So essentially, your brain has specialized regions for memory, emotion, planning, language as well. And these systems kind of work together to make who you are, a thinking and like feeling, communicating human being. That's all. If you have any questions, don't hesitate to put it in the chat. If not, I believe we have a Jeopardy game that we're going to play.
I did see there's one question in the chat about shivering. So just to answer that question, shivering isn't actually related to the basal ganglia because you're right, it's an involuntary movement that's going to be a reflex. That reflex is coordinated, right? So we sense temperature out in the periphery, and it's then coordinated in the brain in different centers of the brain that are not related to our primary motor cortex and that pathway that goes down through the basal ganglia. So basal ganglia is helping us control movements that would originate in that primary motor pathway versus the shivering, it's actually controlled by other centers within the brain, autonomic nervous system centers that help control reflexes like this. And they're actually going to send out other fibers that then modulate our skeletal muscle neurons, those alpha motor neurons that go to the skeletal muscle. They end up getting modulate, their activity is changed by these autonomic centers from the brain that are processing body temperature and cause us to shiver.
If there are no other questions, we can move on to the Jeopardy game. Tim, do you have it open or I could download it? I did not
I
download the
have it
link.
open, yeah.
Okay. So I'm going to open the Excel and keep track. I'm going to first add everyone's names on it, and then I can keep track of the points.
So while we're getting the Jeopardy game ready here, just to keep track, as a reminder, like we said in the beginning, this is a little bit of a friendly competition, and there is a prize for the winner in the winner's school. So make sure that at some point while we're doing this, we're able to get your full name so that we know who you are and where to send the prize for this. And then the way the scoring will work is we're going to end up putting the question up. I'll read the question for you guys and then we'll wait. We'll be monitoring the chat, and the first person with the correct answer in the chat will get those points. And so we'll be keeping track of those points on our end. And then once we've gone through everything, we'll come back, total it up, and see who the winner is. And if there are any questions at any point, again, you can always unmute and ask them, or you can just toss them in the chat, whatever you are more comfortable with.
Okay, I already added everyone's names on the Excel sheet.
Great. We will go ahead and get started then. All right. Should we just go through this in order, top to bottom? Do we want to have people call out
Yeah,
categories and numbers?
yeah, I think people should choose.
All right. Whoever just whoever sends their first preference in the chat is going to be the one that we end up doing. So, what would y'all like to have your first question from? You can also unmute and say it if you don't want to type it out.
Maybe we can start with the intro just to clear it out of the way and get a better idea to the other four categories.
Sure. Intro 100.
Sure.
Okay. All right. These branch-like structures on a neuron receive signals from other neurons. Dendrites. That is correct. All right. So, Love, you'll get the point for those. And then if you want, you can pick our next category in question.
We should do intro into neuroscience 200.
Okay. This lobe of the brain is responsible for high-level cognitive function. What is the frontal lobe? So that'll go over to Ruby for that one. All right, and Ruby, what would you like to do for the next question category?
How about movement for 200?
All right, movement for 200. The large bundle of fibers that connects the two hemispheres, ensuring communication between motor regions, is called this. Corpus callosum is correct. So those points will go to Sammy, and then Sammy, what would you like to do for our next question?
I'll do the first category for 300.
Intro to Neuroscience for 300. These brainwaves have frequencies between 8 and 13 hertz. Alpha waves is the correct answer. So those points will go to Ruby. All right, Ruby, what would you like to do next?
Intro to neuroscience for 400.
Intro for 400. Let's do it. These are the resident immune cells of the central nervous system. If you're not sure, just throw something out there, and that way, once everyone's had a chance to answer, we can go forward if no one gets it. I'm Microglia is correct. So what are microglia? Those are the resident immune cells within the CNS. All right, so that's 400 points for love. And what would you like to do next?
Movement for 400.
Movement for 400 it is. This part of the brain coordinates timing, precision, and motor learning. What is the cerebellum? That is 400 points for Sammy. All right, Sammy, you get the next category.
I'll do senses and perception for 300.
Senses and perception for 300. These photoreceptor cells enable us to see in low light but cannot detect color. Rods is correct. All right, that's 300 points, and you get to pick again, Sammy.
Okay, movement for 300.
Movement for 300. All right, this region of the frontal lobe contains neurons that send signals directly to control voluntary muscle movements. It is not basal ganglia. So the question is asking about where those signals controlling the motor movements originate from. It is the primary motor cortex. That is correct. All right, and you now get the next category, love.
Thinking, planning, and language for 100.
Thinking, planning, and language for 100. This disorder results from damage to parts of the brain responsible for language and affects how a person communicates. What is aphasia? Oh, close race, but love got it by just a little bit. All right. What is next?
Let's do movement for 100.
Movement for 100. In the motor system, signals from one side of the brain control the opposite side of the body. This phenomenon is known as what? It is, well, contra something is not quite correct enough. Does anyone else have any ideas for what it might be? Contralateral control is correct. All right. You get to pick the next category.
Thinking, planning, and languages for 200.
All right. Damage to this frontal lobe area leads to difficulty producing speech, but relatively good comprehension. What is Broca's area is correct, Sammy. So you'll get those 200 points and the next category.
I'll do senses and perception for 400.
All right. This part of the retina is densely packed with cone photoreceptors and is crucial for sharp central vision. Phobia is correct. All right, that's 400 points. And what would you like now?
Thinking, planning, and language for 500.
Okay, going big. The patient speaks fluently, but their speech makes little sense and they have difficulty understanding what others say. This is characteristic of this type of aphasia. Wernicke's aphasia is correct. Nicely done. That's 500 points for love. And what would you like as the next category?
Learning, memory, and emotions 400.
400. All right. This brain imaging technique measures blood flow to detect activity in brain regions in response to different stimuli, including words. I didn't even finish the question. fMRI is correct. Nicely done, Ruby. So that's 400 points for Ruby. And what would you like as the next category?
Thinking, planning, and language for 300.
Okay. Mutations in this gene are associated with language impairments, highlighting the biological basis of language. FOXP2 is correct. All right, so Ruby gets those 300. And what is next?
Intro to neuroscience for 500.
All right, another 500. These chemical messengers are stored in synaptic vesicles. What are neurotransmitters? That is correct again. So 500 more points for Ruby. And where are we going next?
Senses and perception for 200.
Senses and perception for 200. This brain structure relays touch and pain signals from the spinal cord to the somatosensory cortex. Thalamus is correct. All right. And what
I'll
would you like
do
next?
learning, memory, and emotions for 500.
500 it is. Damage to this region, located in the inferior temporal cortex, can result in prosopagnosia or face blindness. FFA. What does FFA stand for? Well, FFA is correct. The abbreviation is for the fusiform face area. Okay. FFA is on the answer slide here, so we'll give it to Sammy.
Sorry, I was like scrambling to write the PowerPoints.
No
So-
worries. So FFA was correct because it's on the answer slide here. So we'll give you the 500 points and let you take the next category as well.
Okay, thank you. I'll do senses and perception for 500.
All right, senses and perception, 500. This brain region, located in the parietal lobe, processes information about touch, pressure, and body position. What is the somatosensory cortex? That is correct. So that's another 500 points to Sammy, and can pick the next category.
Okay, thinking, playing, and language for 400.
Okay, closing it out. This condition involves the inability to understand spoken words despite intact hearing due to bilateral temporal lobe damage. It is not a type of aphasia. So aphasia is an inability to speak. This is an inability to understand spoken words. Any guesses from anyone else? Pure word deafness is correct, Ruby. So their hearing is intact, but they cannot understand words exclusively. All right, so that's points for you. And where would you like next?
Movement for 500.
Movement for 500, the last one. This type of neuron directly connects the spinal cord to skeletal muscles, executing movement commands. Specify the type of neuron more. So motor neuron can technically refer to a neuron within the brain or within the spinal cord, any neuron that's in that motor pathway. So what specific kind of neuron is then taking the message from the spinal cord to the skeletal muscles to cause them to contract? It is not a cortical neuron. Cortical neurons are in the cortex of the brain, and we're looking for a name of the neuron that is in the spinal cord. So synapses occur between neurons where one axon comes near to the dendrite of another neuron, or they do occur between the axon and a muscle fiber as well. So all neurons synapse, and those synapses are the small gaps between one neuron and another neuron, but there's also a synapse where the neuron connects, oh well, almost touches the muscle fiber itself. Love, if you have a guess, you can throw an answer out there, and otherwise we will reveal the answer for this one. Alrighty, so for this one, they're looking for what are alpha motor neurons? So that's the specific name given to those motor neurons that start in the spinal cord and go all the way out to the muscle cell in order to trigger that muscle cell to contract. All right. So before that, I think Ruby was the last one to get something correct. So Ruby, you can go ahead and pick the category again.
Learning memory and emotions for 200.
Okay. This brain structure is essential for forming new declarative memories and was famously removed in patient HM. Hippocampus is correct. Nicely done. All right, what would you like as our next category?
Learning memory and emotions for 300.
All right, learning memory and emotions, 300. These neurons in the temporal lobe respond to specific people or objects. What are concept cells? Ruby beat you by a hair, Sammy. So that is correct. So 300 more points for Ruby, and we're down to just 2 questions left.
Senses and perception for 100.
Senses and perception for 100. These specialized receptors in the skin detect harmful mechanical, thermal, or chemical stimuli. What are nociceptors? That is correct, Sammy. All right, so that's 100 points, and then we'll go to our last question here. Almost. Almost spelled it correctly. There's another C in there. Yes, so this brain region is responsible for decision making, inhibitory control, judgments, and initiating movement. Prefrontal cortex is correct. All right. And then, do we have a final Jeopardy? Do we know?
I don't think so.
Okay. All right. Do we want to then total up the scores and see how we all did?
So yeah, we do have a final winner, and it's pretty close, guys, so good job to all of you, but the winner is Sammy. So
Oh,
good job,
thank you.
congratulations, Sammy. Could you please type in your full name on in the chat box?
Sure.
Thank you, as well as your school. Just, I'm not sure if we have that, just to be sure.
Awesome. So good work, everyone. I think that's all that we had for tonight. So if there any other questions, we're here. We're happy to answer questions. If not, unless Gabriella has anything else, then I think we are all done.
Yeah, I think that's all.